All right, thanks for coming out. I am super excited to introduce today's speaker, Professor Matt Rockman, coming to us from NYU by way of sabbatical from many awesome locations recently. Very briefly, Matt is, by his own words, an evolutionary developmental quantitative geneticist. The way I think about Matt is as a biologist, including one who has studied organisms collected from Chernobyl. Before Chernobyl, Matt did his PhD at Duke and then his postdoc with the Kugliak group. And he has been running his own research program for a bit over a decade now. And that's the context in which I first met him. And aside from being a huge fan of his research, one of the reasons that I really look up to and I admire Matt's work is not only has his work kind of laid ways forward for the field, but at increments that felt very necessary, it's given us the tools and the foundations to do self-reflection as evolutionary geneticists. So as methods and new things develop, many of the fundamental questions remain the same, and yet the way we interact with them change. And more so than anybody else I can think of, Matt has really helped clarify those tensions and opportunities that exist in the field. For example, many of us seek to map traits to genetic variants. But when we use that approach, what are the questions that we're actually able to address with the genetic variants we find for the purposes of evolution? A different concept, for example, pleiotropy. It's an organizing principle that many of us allude to in our research. Well, what do we actually mean? And does an evolutionary geneticist and a molecular biologist mean the same thing when we refer to pleiotropy? And in clarifying those definitions, or letting them remain different, what do we gain? And also, what do we lose in the confusion of them? But today he'll be talking about Evo-devo, and very briefly, I think kind of just the clarity of ideas that Matt has and the way he can put them to words is best articulated from his own writing. Describing his lab's work, Matt wrote that we study the relationship between the molecular, cellular, and developmental mechanisms that generate phenotypes, and the population and community level factors that shape variation of those phenotypes. This is Evo-devo, and today I'm excited to learn about how Evo-devo is a PopGen problem.
Thank you. Thank you all for coming out today. I'm really excited and happy to be here and to tell you about Evo-devo as a PopGen problem. And I'm a little bit concerned that I picked a kind of controversialist title here, and also that I've had an introduction from Mo, and now expectations are in the wrong spot. So this is going to be me trying to articulate the idea that that Evo-devo has something to teach to population genetics. And it's really motivated by this kind of long-standing question that Evo-devo has been pushing for a long time, that the synthesis that created our modern understanding of evolutionary biology missed something by leaving out development. And a lot of people believe that the synthesis cannot be complete until it's integrated development. And population geneticists, I think, rightly say, actually, our equations don't need that. The equations work just fine without thinking about development. We can just think about alleles, we can think about genetic covariances in a kind of quantitative genetics framework. We don't need to know those details. And so the thesis that I will be advancing today is that in specific contexts, when we're studying the evolution of developmental phenotypes, actually those equations are wrong. And that if we don't incorporate certain facts about development, then we're going to get our ideas about how evolution works wrong as well. So that's the thesis. And to illustrate these claims, I'm going to talk about one of our experimental organisms, this amazing little polychaete annelid called Streblospio benedicti, which is one of the model systems that we work with in the lab. Most of my lab works on C. elegans and its close relatives, and those are incredibly powerful genetic models for lots of questions. They're the easiest animal by far, C. elegans. Genetically, you can't beat it. We can freeze them and thaw them. They have a 2.5 day generation time. It's magic. The thing that they don't have is phenotypic diversity. They all look exactly alike. They all have exactly the same cells. So they're not a great model for these kind of big macro Evo-devo questions. This animal is a great model for that, and that's why we studied. I'm going to tell you a bunch of the details about how it works, but first I want to introduce the folks who have done most of the work I'll describe today. Christina Zakis and Connor Gilligan. Christina was a postdoc in the lab and really spearheaded a lot of our early work. She's now got her own lab at North Carolina State. Connor Gilligan is a PhD student in the lab right now, and he's been pushing the system into population genomics kinds of approaches. We've also had help from an undergraduate and a master's student, Jen and Alex, and of course we have funding from a variety of sources as well. So this Evo-devo problem I'm going to talk about, I want to start in the place in nature where Evo-devo is kind of, I think, playing out most dynamically, and that is on the seafloor. And we're going to focus on these benthic marine invertebrates. The polychaete that I'm going to talk about is benthic marine invertebrate. So are these sea urchins. These are animals that live as adults on the seafloor. And most such animals, which cover, by the way, a huge piece of animal diversity. If you look at the phylogeny of animals, many different branches have benthic marine invertebrate lineages. And so this is a very common way of being an animal, adults on the seafloor, but the offspring are often swimming around in the water column. And so these animals will make planktonic larvae that have to feed, they have to evade predators, they have to go through metamorphosis and then come back to the seafloor to become these benthic adults. And many, many different lineages have evolved unique and distinctive larvae. So here's a figure from a paper from Gunnar Thorsen in 1950, where he shows these 5 different examples of larvae of benthic marine invertebrates. So this is on the left we've got, this is a sea urchin. It's going to make a little out-pocketing that's going to turn into a sea urchin, and then it'll fall over on its side, and the sea urchin will crawl away, and that larva is lost. So a radical metamorphosis. And so that's the sea urchin. This is a sea cucumber and a sea star. This is a ribbon worm. This is a polychaete. And these animals are going to swim around and eat and go through metamorphosis. But in each of these separate lineages, there have been evolutionary transitions to direct development. And so this is a sea urchin and a sea cucumber and a sea star and a ribbon worm and an annelid. And these animals are hugely larger. So each of these is in life 5 times larger than it appears relative to these other guys up here. And these animals do not need to feed in the plankton. They can undergo the metamorphosis without ever acquiring nutrition from the sea. Instead, they use the yolk that was provided by their mother. We call these different life histories planktotrophic, they eat plankton, and lessotrophic, they eat yolk. And so transitions from planktotrophy to lessotrophy have happened hundreds of times across animal phylogeny. It's one of the most characteristic kind of macroevolutionary transition repeated across phylogeny in the animal world. And so this is something that if we could kind of understand what's special about what's going on here, we can understand a kind of a thing that's common in evolutionary history. And the thing that is most important to think about the genetics here is that the differences in size between these guys is governed by the size of the egg. And the egg is a phenotype not of these larvae, but of the mother. And so to evolve from something like this to something like this, there have to be 2 kinds of changes. There has to be a change to the provisioning of the egg, and there has to be a change to the zygotic larval developmental program that's going to decide what the phenotype of that egg is going to turn into. So we've got maternal and we've got zygotic. And this is pretty important for the genetics. And so I just want to remind you a little bit about what's special about maternal effect genetics. Here's the idea that individual mothers are genetically different from one another, and those genetic differences will contribute to phenotypes of their offspring independent of the genome that the offspring inherit. So the offspring will have a zygotic genome that gets a random half of the genome from the mother and a random half from the father. That's a new diploid zygotic genotype. But there's also a phenotype of that zygote that is governed exclusively by the mother's genome, her diploid genome, even the alleles that she's not passing to the offspring. And so this has special genetic properties. One thing is that maternal genetic effects are sex limited in their expression. Only the mother is producing these traits. It's also that selection on these maternal effect traits is quite indirect because the phenotype that matters belongs to an individual different from the individual whose genotype matters, and so selection is weakened as a result of that imperfect correspondence. But probably most important, and kind of least intuitive, is that we end up with these zygotic genotypes here occurring in different kind of maternal effect environments as a function of the frequencies of these maternal genotypes. And this creates, sorry, here's the punchline. This creates frequency-dependent genotype environment covariance. In other words, for example, the little a allele is going to occur in these different backgrounds as a function of the genotypes up here, which are a function then of Hardy-Weinberg segregation in the previous generation. And so particular alleles find themselves more often in mismatched genotypes than in matched genotypes if the kind of matched genotype is rare in the population in the previous generation. So this requires then that if we're going to talk about the evolution of developmental phenotypes like larval morphology, then we have to incorporate not just the genotypes from one generation to the next, like a normal recursion equation, like a typical population genetic model, but instead we have to include two different generations and frequency-dependent genotype environment covariance, which sounds very esoteric, but the point here is that any organism that develops from an egg, just to say all animals, is going to have early developmental phenotypes that require this model. A simpler model is not going to capture the dynamics correctly. So I'm now going to show you that this is true in the case of our model organism Streblospio benedicti. So this is an annelid segmented worm like an earthworm, and it turns out to have both of these kinds of life histories, lesthotrophy and planktotrophy, segregating within a single species. And this was discovered by Lisa Levin back in the early 1980s, and she published a beautiful series of papers through the early 1990s showing that this was a heritable feature of these animals. And then she switched fields, and we've been picking it up now some years later. So just to remind you, annelids are part of this branch of animal phylogeny called Spiralia that includes an enormous piece of animal diversity. All of the mollusks, all of the annelids, all of the flatworms, and then a bunch of kind of less familiar things, brachiopods in there, ribbon worms. Huge number of species, very different kinds of organisms, 0 model organisms in that entire clade. Down here in the ecdysozoans, we've got our C. elegans, we've got our flies. Deuterostomes have mouse and zebrafish and so forth. But we're really operating in a bit of diversity here where we don't have a lot of resources to lean on, so we have to develop a lot of tools. The kind of fundamental biology of these animals is that they have separate sexes, males and females, and they have internal fertilization, and then the females will brood embryos in little pouches on their back, and I'll show you more about that. But first I'll just explain this kind of fertilization business because it's unusual. The males produce spermatophores and then just deposit them on the seafloor, and females come along and pick them up and internalize them, and then they can store sperm and use them for months. And I've got a little video I made here. This is an animal in this little mud tube, and this is a male. It's waving around its palps, which it uses to collect food. It's got these two structures here called branchii, which are mostly gills, but the males also use them to kind of projectile throw the spermatophore onto the substrate. And that's what's happening here, is that this male is using cilia on its branchii to throw a spermatophore. There it goes. And then it'll retreat back into its little tube. All of this stuff in the surrounding area is worm excrement. And so I think we can all agree that it's a very charismatic animal. Here is a view of a piece of a female from the side. So the animal's head is over here, and here's the gut running through the middle. And inside the body cavity, there are these oocytes here, waiting to be fertilized. And this is a lesthotroph. It's got these great big eggs. And after fertilization, it transfers the zygotes to brood pouches on its back, and then the larvae go through embryogenesis in those pouches. And so here are some lesthotrophic larvae just about to be released into the water column. And so in both the planktotrophs and the lessotrophs, all of that kind of cleavage stage gastrulation stuff, that's all happening in these protected brood pouches. It's only when they've fully developed into little larvae that they're released. And at that point, these lessotrophs can immediately go into the mud and be juveniles, but the planktotrophs have to then swim around for, in some cases, weeks, eating plankton and putting on mass. And so those animals look pretty different. So here's a lessotroph, and here's a planktotroph. And you see, anatomically, they're different. These guys start with many more segments. These guys have to eat to make those segments. These guys have these defensive bristles, and they have these cirri on the back. And these guys don't have any of those structures. And these structures are larva-specific. So at metamorphosis, those are thrown away. It's also important to notice that the lessotrophs, the mothers, make many fewer eggs than they would if they were planktotrophs. And those eggs are much larger. And so it's basically like an 8-fold difference in these things. And I'll just point out that these structures here seem like they're probably useful. And we know that from studies in another annelid called Platanarius. I'm going to show you a video. This is a study that was investigating neural control of their behavior. And here is a wild-type Platanarius larva being attacked by a copepod. And you might have seen that larva flared its bristles, and the copepod was unable to capture it. In this study, they then ablated that ability from the larva, and there's the result, is the copepod eats it. So these appear to be defensive structures to keep them from being predated. These animals live all up and down the east coast of the United States. That's their native range. They also occur as introduced species in a number of estuaries around the world. We can collect them right in New York Harbor. So this is Manhattan up here. This is NYU right at the corner here. This is Brooklyn. This is Staten Island. Does anybody know what this is? That's New Jersey. Hudson County, New Jersey, right there. And so here in Bayonne, New Jersey, this is Newark Bay over here, and we collect planktotrophs right here. And over here in the New York Harbor side, we have a site that contains mostly lessotrophs, but also some planktotrophs. So we can collect these guys. Here's actually a PhD student, Jackie Jackson, collecting some polychaetes from Newark Bay. They're in mud everywhere on the east coast of the United States at incredible densities. So they can be tens of thousands per square meter. They're like maybe 2 centimeters long as adults. So they're pretty small, but they really do dominate the mud flats. So this is not a super obscure animal. It's a super obscure animal, but it's a very abundant, widespread animal at the same time. We can raise these in the lab in 6-well culture dishes with artificial seawater. They live in some terrible places like Newark Bay, which is right downstream of the Passaic River and the Hackensack River, which have Superfund sites. And so they're living with horrible stuff. And they're also living on mud flats that are sometimes exposed at low tide. So they can put up with salinities, they can put up with temperatures, they can put up with toxins. They're great lab animals because they don't care about that stuff. And so we can grow them with artificial seawater and mud that we just collect from Newark Bay and then sterilize and put in these dishes. So here's just an example of one of these dishes that has one adult worm in it, and that worm is going to make a mud tube, and here's the mud tube, and then it's going to eat and defecate, and then sometimes it'll turn around in the mud tube, and then eventually it'll make a new mud tube, and they just go through all of this mud and reproduce, and it's great. So here's some things that we know about these animals' evolution. We know that the larval form is highly heritable and not plastic, and there's a bunch of studies that have shown that. And we know from common gardens in our own lab over many years that lessotrophs make lessotrophs and planktotrophs make planktotrophs, and so forth. We know that typically localities are homogeneous. They just have one type or the other. So if you go out to a mud flat, then probably all of the worms are going to be planktotrophs, or all of the worms are going to be lessotrophs. And there are some exceptions. We've already seen one in New York Harbor, but most of the time a mud flat is one thing or the other. And there's no cline along the coast. It's not like lessotrophs in the north and planktotrophs in the south. It's mud flat to mud flat. We also know that populations are linked by gene flow, and we've done a lot of population genetics, and I'll show you some more, that most loci in the genome are not differentiated according to developmental type. So it really is one species. So having introduced this organism, I'm going to tell you the kind of game plan here, which is that we're going to write down a model for the fitness of these animals. This is kind of a theoretical link between phenotype and fitness. Then we're going to talk about the empirical connection between genotype and phenotype, that's linkage mapping and association mapping and so forth, where we kind of figure out the number of loci and how they affect the different traits. And then we're going to ask, can we put together genotype, phenotype, and fitness and figure out what the implications are for how these phenotypes should evolve in these populations? So now I'm going to quickly introduce a kind of a life history model that's based on a classic model for the kind of trade-off between the number of offspring and the size of offspring. And in most of ecology, there's the Smith-Hartwell model, Fretwell model, that describes this. But for historical reasons, marine larvae people have a different model that was developed by Rick Vance in the 1970s. And it's kind of an amazing thing where this is exactly the same question, trade-off between number and size, but those two literatures are completely missing each other for the last 50 years, which is weird. At any rate, the model goes like this. You could have some kind of model that's going to tell us how many offspring will a mother have that successfully reach adulthood, or at least metamorphose to being the benthic adults. And so that number here is how many offspring that mother's going to have. And that's going to be a function of the total amount of maternal investment and the size of the offspring. So if we imagine that a lesotroph, an animal that can immediately metamorphose, has a size of 1, right here, then our planktotrophs have a size of 1/8 of that. By volume, they're 1/8 as big. And so the number here is going to be 8 times as many. So if we imagine that maternal investment is fixed for everybody in the population, then the number of offspring is just a function of how big those offspring are. That's so far so good. So we're just going to abbreviate that here. Maternal investment, size of the offspring. But the key here is that that in this case, the planktotrophs would always be better. You're going to have many more offspring if you make smaller offspring. But those guys have to swim around in the plankton where they're going to get eaten by predators. And so how many of those planktonic guys are going to make it to metamorphosis? Well, we can imagine that there's this constant risk of getting eaten by predators. And so the number will decay exponentially as a function of the time it takes to reach metamorphosis and the predation rate. And so how long is it going to take to get to metamorphosis? That's really going to depend on how big they are. Right? This guy is going to take 0 time. These guys are going to take much longer. And so if we imagine that that's kind of a linear relationship, then 1 minus the size is going to tell us how long it's going to take them to get to metamorphosis. There's some kind of scale that tells us the time to metamorphosis that is variable as a function of how big the offspring is. Meanwhile, there's a predation rate. And that predation rate is going to depend on how many predators there are, but also on how well the offspring can resist those predators. And so you've seen that those defensive spines can deter a copepod. And so those guys that have defenses are going to have a lower predation rate than those guys that don't have defenses. And so that's another parameter that we should add to this model. And we're going to say, well, there's some kind of maximum amount of defense that an organism could have. Spines are going to defend against a copepod, but they're not going to defend against a whale. And so there's some maximum amount of defenses. And then the amount toward that maximum is going to be a thing that varies as a function of genotype, and that's this defensive investment here. So then we can describe our lesothotrophs here. No defenses, size to develop to metamorphosis without feeding. Over here, these guys have maximal defenses and are 1/8 as big. If we put this into a model now, the thing that we find is that everybody, no matter what the conditions are, is going to have defenses, because there's no cost to having defenses. But in nature, we know that, in fact, the lesothotrophs don't have those defenses. So there must be a cost, and we should put that in the model too. And I will be honest that we don't exactly know how it should go into the model. And so we've done it in the stupidest possible way, which is just by saying that if a mother is investing in defenses, then she's going to have fewer offspring. And so it's just kind of tacked on here at the end. So there's some cost of maximal defenses, and then each individual's investment is scaled by however much it's investing. That's the same D here, 1 and 0. So that is our model. And some of the terms here in this model are features of the environment. How long does it take to get to metamorphosis? That depends on how much plankton there is for the planktotrophs to eat. If it's real sparse, they're going to have to swim around for a long time to find one cell of algae. If there's a bloom, then they can just eat and eat and eat and metamorphose. So that's a function of the environment. Same as this predation rate here, how many predators there are. That's not up to the polychaetes. On the other hand, these things are all features of development. And so those are things that the genotype of the worms could influence, and those are things that can evolve in response to the fitness landscape here. And so we're going to assume, for convenience, basically that all of the worms are investing the same amount in offspring. So we're going to kind of fix this total maternal investment. And we're going to suppose that the cost of building defenses is also constant. And so the things that are going to vary in this model are how much is invested in defense and how big are these larvae relative to a lestotroph. And so those are the things that can evolve. And so here's what our kind of phenotype space looks like. We've just got these two things here that can vary, defensive investment and egg size. And now we can kind of take this phenotype space and add a fitness axis. And so that's what we've done here. We've kind of tilted this on its side and added a z-axis that's going to tell us relative fitness. And now we can let the environment vary and see which phenotypes are most fit. And so here's a little thing where I'm going to let these things move around, and we'll see what happens to the fitness landscape. It's time to metamorphosis, varying, and predation rate, and so forth. Everything can vary. But no matter what varies, a couple features stay the same. Only two spots are ever the maximum fitness, and that's the lestotroph and the planktotroph. None of the alternate combinations, none of the intermediate states are ever optimal. So there's just these two opposite corners that can be the optimum one or the other. When the environment changes and one that was optimal becomes suboptimal, there's never like a ridge that connects that phenotype to the opposite corner. It's always got this depression in the middle. So there's never a smooth way to get from one corner to the other. It's always this jump across this reduced fitness spot in the middle. So those are really the key takeaways from this, is that the two forms that we observe are the forms that our model predicts should exist. But evolutionary transitions should be really difficult because all of the intermediate states are suboptimal. All right, so that's the phenotype to fitness model. There's no data there, but that's the way we can kind of conceptualize how this could work. But then what happens when we add genetics here? Well, it depends on what kind of genetics we imagine are underlying the differences between the planktotrophs and the lestotroph. So we could have a super gene, like maybe there's a master regulator that switches the whole thing. Maybe there's a whole bunch of genes that are linked in an inversion that segregate as a single unit. That's actually a thing that's observed quite often in this kind of dramatic difference between morphs within species. So we can imagine that there's one gene that moves from one corner to the other. But we can imagine any number of other architectures as well. We could imagine that there's two loci. Or we could also imagine that there's two loci, but each one only affects one trait or the other. So they might or might not be pleiotropic. And similarly, we can imagine kind of more polygenic versions of this. I've written 40 loci here, but of course, polygenicity could be 1,000 loci affecting all of these things, and they could be varying in size, and they could be some of them pleiotropic and some of them not. And over here is the actual genetic architecture of our animal. And so that's the kind of space that now we want to think about. Do the details of this architecture matter for how things would evolve on that fitness landscape? But we need one more dimension here, which is that we're pretty confident that egg size is going to be a maternal effect phenotype of the offspring. That is, that in offspring, larva size is governed by how big its mother made its eggs. That's a maternal genotype that matters for that trait. On the other hand, the defenses conceptually could be maternal or zygotic. And so when we think about these models, we can think about a model where the mother's genotype determines what the larva looks like, or we can imagine a model in which these two things are separate, where the mother's genotype governs the one axis and the larval genotype governs the other. And I'll just mention that it could be a single locus that acts maternally and also zygotically, right? These don't have to be separate loci. They could be, but we can have one supergene that acts maternally on one trait and zygotically on another. These have implications for how these traits will vary in populations. So if we imagine a purely maternal model, where here this is a planktotroph, its genotype at a supergene would be big P, and over here we've got a lestotroph, it's a big L. And in this case, the offspring just all look exactly like their mothers. So that's pretty straightforward. If we have a maternal zygotic model, then we get a situation where the offspring have a size that's the same as their mother, but their larval phenotypes are now going to be the joint product of the maternal genome and the paternal genome that are present in the zygote. And so if it's additive, for example, then everybody's going to have half-sized defensive structures. And if we go another generation, then we're going to get situations where now the zygotic traits are segregating, but everybody within a brood has the same mother, so they've all got the same size. So there's these differences in the amount of variation that would be generated and how correlated they are with the maternal traits. So that's just kind of illustrating the complexity of a situation when you have maternal and zygotic traits. What's the reality for these worms? Well, the first way we tried to figure this out was to do old-fashioned quantitative genetics line crosses. And we crossed these guys, and F1s, if their mother is a planktotroph, like here, then all of the F1s will be small larvae, because the mother is making small eggs, planktotrophic eggs. And so that's what's going to happen here. Then those guys grow up to make their own offspring. And what we observe is that these guys, these F1 mothers, make intermediate-sized eggs. So they're heterozygous at every locus that affects egg size. And it turns out it behaves more or less as an additive trait at that level. The offspring are intermediate in size in that next generation. And these guys then, the F2s, grow up as intermediate-sized larvae. When they make eggs, these females, they make diverse-sized eggs. Every female makes only one size of egg. The brood is all is uniform, but some mothers are making big, some mothers are making small, some mothers are making all sorts of grades of intermediate egg size. And already in this generation, the defensive phenotypes are segregating. So that's telling us that those are zygotic. And so that's from just classic line cross genetics, looking at the distributions of phenotypes. And we did another generation here to look at the segregation now jointly of the egg size in this generation to make these larvae. You have to measure the G3s to know the egg size phenotype of the F2s, and also the zygotic traits. And for this, we then did quantitative trait mapping to figure out which loci are affecting which traits and so forth. So here's again a picture of a planktotrophic larva. And it's small. They've got 4 eyes, which are not all in focus here. And they're kind of classic polychaete larvae, which have ciliary bands that they use both to swim and to capture food particles. And these guys have these larval bristles that are actually serrated, and they can flare them. And they've got these bacillary cells, which are these giant things that have these little rod-shaped inclusions and stick out. And so these things are called anal ceri. And those are unique to the planktotrophs. The lestotrophs don't make that cell type at all. We don't know what those cells do. Nobody does. But they're unique to this phenotype. So we did this joint analysis of 264 F2s. I should say that Christina Zakis did these crosses, raised these animals, genotyped them. This is Christina Zakas's work. And then 3,954 animals in the next generation. And then we can fit these beautiful quantitative genetic models that say, well, what would we observe if this were maternal? What would this look like if it were zygotic? What if it's both? And so forth. And so we can separate out which effects are maternal and which effects are zygotic from the phenotypes of these animals in these multiple generations. And this analysis is published. And I'll just point out that, that you can work on C. elegans all day long and never get a journal cover because they're so boring looking. But people love a polychaete. OK, so here's the first punchline, is that the anatomy of the larva, the length of these bristles, the number of these bristles, and the presence of these cirri back here, those are purely zygotic. The maternal genotype does not matter in any way, except insofar as the alleles are transmitted and the genotype of the zygote is doing the work. And so you'll see that we have a modest number of loci, and these loci are specific to specific traits. So for example, this locus is the number of these bristles, and this locus is their length, and this locus does not affect length, and this one does not affect number. And so there's pretty discrete phenotypes happening here in a modest number of loci. And the effect sizes are quite substantial because we can just see them looking at these plots. So for example, this is for the two loci that affect chaetae length. This is the double planktotroph homozygote. It's got lots of, it's got long chaetae. Here's a lesotroph. These guys have short chaetae, and many of them have 0. And then if you add incrementally additional alleles, then you kind of step up. This is stepping up within this locus, and this is stepping up in the other locus. And you'll see that these are substantial effects for all of these traits. When we look now at the size of the larva, we're looking now at the size of the G3 larvae, which is a function of the F2 maternal genotype. We see here that there are no zygotic effects at all, which is as you would expect. It's governed entirely by the genotypes of the parents. And we've got, again, a couple of loci. This is a kind of typical non-model QTL situation where we're not getting a lot of resolution beyond the chromosome. The spatial detail within a chromosome is not very good, but we can find associations with individual chromosomes. A nice thing about our statistical model is that we can distinguish maternal from paternal. And typically, we're using paternal as kind of a negative control. We wouldn't really expect to see that. And to our surprise, 2 of these loci show significant paternal effects, where the genotype of the father matters, and at the same locus, the genotype of the mother matters, but the genotype that's actually inherited by the zygote doesn't matter. So purely parental. And I'll just show you what that looks like kind of phenotypically. Here's a typical maternal effect locus where each additional lesotroph allele makes the offspring larger. That's what we expect to see. So that's the typical situation. And here's another one just like that. At these 2 other loci, the parental genotypes matter, and they matter in kind of similar magnitude but opposite direction. So the paternal genotype is kind of canceling out the maternal genotype. We don't know mechanistically how this works, because the phenotype is the size of the offspring, and so it's not obvious how a sperm would influence that directly. One possibility is that the females are picking up spermatophores and internalizing those, and those are chemically communicating with the female germline in a way that affects the development of the egg. So it's really, the physiology is on the female side, but the genotype is the paternal, and it's transmitted through the spermatophores chemistry. That's just a hypothesis. So having identified that there's totally separable maternal and zygotic effects, and that these are trait-specific and so forth, then we want to go a little bit further and find, can we actually get down to loci so that we can say, well, there's actually this many loci, and this whole chromosome that looked associated is actually just one locus, and so forth. And to do that, we've turned to population genomics. So first we had to make a genome, and here again, to reinforce my point, it's so different to work on an animal that has morphology. So we made a genome using all of the everybody's favorites, long reads, and a linkage map and high C. And it's good. It's got 10 autosomes and an X chromosome. And we've got karyotypes that match that. And that's all good news. And now we can go out and just sequence a bunch of individuals who are plankotrophs and a bunch who are lescotrophs and look for what alleles differentiate those two classes of animal. So we went back and collected our guys in New Jersey. And we also collected a kind of matched set in New England. This is Providence, Rhode Island here. This is Fall River in New Bedford. This is Woods Hole, Massachusetts right here. And we got plankotrophs over here in Rhode Island and mostly lescotrophs, but some plankotrophs down here in southern Massachusetts. And this is work by Connor Gilligan. Here's Connor in southern Massachusetts collecting some animals. This is a much nicer spot than Newark Bay, New Jersey. So he got these animals and sequenced them at high coverage. So he's got 48 animals total. The genome size is 700 megabases. He sequenced them all at 30x coverage. And he observed about 38 million SNPs across the whole collection of 48 animals. And now we can ask, how are those distributed across phenotypes and geography? And here's kind of the first big takeaway, is that there's a huge difference between the plankotrophs over here. This is a principal component analysis of the genotypes. That the first principal component of the genotypic variation is the morphology. So plankotrophs over here and lescotrophs over here. And there we go, there's the labels. And the plankotrophs all kind of cluster together in both axes here. So there's a black triangle under here. We have not very many plankotrophs from the mostly lescotrophic population, but they look like perfectly good plankotrophs in this genotype space. There's no signature of them having been mixed with lescotrophs. And these guys are all really close together. The lescotrophs over here are really far apart in principal component 2. And so this, I think, is telling us something about the downstream consequence of the life history evolution on population biology, and that is lescotrophs settle where they're born. They don't migrate, whereas the plankotrophs are out there in the ocean for sometimes weeks and are traveling all over the place. And so this kind of couples life history and population biology and spatial genetics and so forth. And I think this is a thing that was predicted, of course, and earlier data didn't actually show it for reasons that I'll get to in a second. So this is the first time we've actually seen this signature that we think should certainly have been there. So when we look genome-wide at these 38 million SNPs, one surprise is that this differentiation between plankotrophs and lescotrophs doesn't amount to much across the genome. So here is the differentiation, the distribution of a differentiation statistic, FST, between the plankotrophs and lescotrophs. So we're ignoring geography now. This is just all the plankotrophs versus all of the lescotrophs. How differentiated are those two pools? And the answer is not really at all. So the FSTs are down here. The mean is 3%. And so indeed, most of the genome, it seems, doesn't care about morphology. It's moving back and forth between these two morphs indifferently. What if we look at our loci that we know are associated with these traits? So here again is the genome along the x-axis here. And I'm just showing you which traits mapped to which chromosomes in our QTL analysis. Now we can ask, are there FST peaks on those chromosomes? Because then we would say, well, that's the peak that corresponds to this trait, and that's the peak that corresponds to that trait, and so forth. And the answer is yes, there are peaks for those chromosomes, and there are also peaks everywhere else. So this is a plot that it's cut off actually at the bottom here because almost all of the mass of points is down here below 0.1. And so there's actually not that many points up here at these high FST peaks, but there are some. And I seem to have lost a slide here. Sorry, let me find this one again. There's 38 million SNPs that are analyzed here. There are 5,140 that have an FST of 1. Perfect association with the phenotype. And so those are points along the top. Each of those points along the top is actually multiple SNPs in a little haplotype block. But there's 5,000 or so of those. Those include about 26 non-synonymous mutations. So those are good candidates for being causally implicated and stuff. If we zoom in here on just chromosome 3, and now I've cut it off at 0.5. So all of the points here represent the most differentiated 0.3% of SNPs on this chromosome. And you can see that there are multiple discrete peaks along this chromosome. So we're actually not currently able to say this is the peak that's this trait and that's the peak that's that trait. But what we do know is that there's more peaks than there are traits that we measured. And that's because we measured kind of the obvious stuff, the size of the offspring and the presence of these chetae and the serae. And those are things that are just easy to measure under the microscope. But there's all sorts of other traits that also have to be different in these morphologies. And so if we just look at the genes that are under these peaks, we can get some clues. And one clue is that a lot of them are involved in stuff that plausibly has to do with the quality of the yolk, which is obvious in retrospect, I should say. We didn't measure any features of the yolk. We can do lipidomics now and figure out if these are meaningful associations. I'll say that in the lab, the F2s, the G3s, they're all totally fine. We don't see like kind of intrinsic incompatibilities between these genomes. We don't see those larvae having more trouble settling. So I think if we put them in lab conditions and just cover them with food, these details don't matter, but in nature, presumably, they're very, very important. So that's kind of what we get from a first glance at the population genomic data, which are pretty new. And now we can ask, what does this mean for how these traits are going to evolve in populations? We considered a single locus or two or many. And now we know that the reality is down here somewhere in the many category. And so we can ask, well, what would happen if a population experienced a change of environment? Could it switch from one morphology to the other? And so we're going to imagine a population of lesthotrophes down here, and into this population comes a planktotroph. If the environment changes now so that planktotrophy is favored, can planktotrophy invade this population? So we're going to address this with simulations, in part because once you get to lots of loci and maternal zygotic interactions, the number of recursion equations required in a system of, the number of equations required in a recursion system is thousands, and it's not tractable. So we're using simulations. And so we're going to suppose that the adults live in a mudflat, and that has a carrying capacity, and there's going to be random mating, and each mating is going to yield some number of progeny that's a function of the genotype of the mother, and the larva's genotype is going to determine its investment in defenses, and that'll vary as a function of which genetic model we're going to use. And then there's larvae settle, and that's given by the relative fitness from the phenotype to fitness landscape model that we talked about before. So that's this model here. I'll just mention that this model is telling us basically the fitness that's expected under particular conditions for a genetic clone, where everybody of that phenotype is going to have offspring of that phenotype that exactly matches it. Once we add segregation of genes, then the fitness of a particular phenotype no longer depends exclusively on its phenotype. It also depends on the genotype and the genotype of others in the population as a function of this maternal zygotic interaction. So let's suppose that we start with our lesthotrophes in the mudflat. Lesthotrophy is the favored phenotype here. And we're going to change the environment. So now there's a shorter time to metamorphosis because there's a bloom of algae for the planktotrophs to eat. Now planktotrophy is radically favored up here. What's going to happen? So we'll start with this supergene model, which we already know is not the true genetic architecture, but it's kind of illustrative. We're going to start with lots of lesthotrophes and see what happens over evolutionary time. The fitness maximum under these conditions is down here with the lesthotrophic allele frequency of 0. So if we were allowing clones to evolve, then we would see a quick drop down to 0 frequency. In fact, if we let this supergene evolve, then what we see depends on the genetic architecture, and in particular, the presence of an interaction between maternal and zygotic. If we have a maternal-only model, then indeed planktotrophy can invade. If we have an interaction between maternal and zygotic, then it can't, and there's this frequency dependence that traps the population at this lower fitness peak. So it can't escape this low fitness situation because of the genetics, not because of the fitness landscape. So this is kind of unintuitive, and we're going to walk briefly through how this works. So we're going to show individual genotypes over time in a maternal-maternal model and a maternal-zygotic model. So here's 90% of our population has big eggs and no larval defenses, they're lestrophs. 10% is planktotrophs, lots of defenses, low size. And here's the population average. In the next generation, here's what happens. In this maternal zygotic situation, the offspring are the same sizes as their mothers, but the defenses have now segregated so that we get these intermediates in the F1s. It hasn't segregated, it's that the F1s have this intermediate phenotypic value. And one thing you'll note is that because of random mating, this retains a huge fraction of the population. That's lestrophs that have mated with lestrophs. Some of the lestrophs mate with planktotrophs, and they make these guys over here. But most of the lestrophs retain this corner. It's a different story for the planktotrophs, because they're rare. And that means that planktotroph matings are also rare. Most of the planktotrophs mate with the lestotroph. And so the bulk of this population has ended up over here in this maladapted bit of space. That didn't happen over here because the offspring look just like their mothers. And so we end up still having lots of these high fitness planktotrophs here. And in fact, the proportion has gone up. This average dot has moved a little bit toward the planktotrophs because they have higher fitness. And this is going to continue. That's the kind of takeaway, I think. This is going to continue. We get more segregation. And so now there's really very few over here, even though this is the highest fitness, whereas this group is accumulating and so forth. So if we just watch this as a video, then we see that the maternal-maternal has a whole different result from the maternal-zygotic. This is the supergene case. We know that the reality is polygenic. There's lots and lots of loci involved. And so to kind of make a long story short, polygenicity is terrible for this situation. It really it reinforces the degree to which these animals are trapped by positive frequency dependence. And so the genetic architecture here means that when we go out on a mudflat and see planktotrophs, that's not actually giving us a very good reason to expect that planktotrophy is the optimal phenotype for that environment. That it could be very much dominated by kind of priority effects. If you have an empty mudflat, then whatever worms get there first, they're going to be the worms that can hold that space just by virtue of this positive frequency dependence. So in that sense, if you're studying a phenotype that involves early development, and you're just thinking about it in terms of, Fisher, for example, or classic recursion equations, those equations are going to give us the wrong answer about how fitness is evolving in these populations. We need to take into account this genetic architecture of these traits. And I'll say that population genetics already has those equations. They're just much, much more complicated and not the ones that we go to first. And so this is saying that for this class of phenotypes, we have to go to those equations. And that's going to help us understand what's going on in this kind of macro scale, where this particular transition somehow has nevertheless happened a whole bunch of times. And that part is where I think actually identifying molecular genes is going to help us understand what has to actually happen developmentally to get a lestotroph from an ancestral planktotroph. And we're not very far on that yet. And with that, I'll thank you all and take any questions.
Curious on a sort of macroevolutionary scale. You seem to imply that, and I'm going to lestotrophs is the derived state, and planktotroph is the ancestral. Are there situations where it reverts back to planktotrophs?
Right.
How labile
Yeah.
in both ways?
Yeah, that's a great and I would say not satisfactorily resolved question. So people applying like just the most kind of basic phylogenetic inference to the distribution of the trait infer a couple of reversions. But I think that it really depends on your kind of prior about which one is going to happen more often to trust those. So I think it's unclear right now.
At the beginning, you mentioned that benthic invertebrates is such a large portion of the animal kingdom.
Mm-hmm.
What is the benefit to lesotropes? Like I know they start off larger, but if they remain on the benthic floor, but they lack the defenses of the planktotropes, what's keeping them from just being easily predated upon?
Oh, right, yeah. So the predation in the model is entirely in the plankton. So we're imagining that once an animal gets into the benthos, into the mud, then in the model at least, we no longer follow it. But if we did, we would suppose that it would be the same for the planktotropes and the lesotropes at that point. That now that they're juveniles, they're all experiencing the same predation regime, so that's no longer going to discriminate between them. Yeah, but I think the basic model for a lot of these benthic animals is that plankton is extremely dangerous as a way of living, and that they're better protected from predation in the benthos.
Okay.
Please.
So you mentioned that they pick up spermatophores from the ground. Is there any mate choice that happens? And
right.
how does that affect?
Yes, right. So that seems like the thing that should happen is that because these hybrids are all going to be mismatched, that there should be very strong mate choice and assortative mating. And recently, Christina Zakis' lab collected and published the first data on that question. And they got a resounding yes, that there is very strong assortative mating in this laboratory condition in which each female is choosing between two males. And so that it's clear that that's possible. And sort of one sort of remaining question about that is that those experiments involve involve mate choice between animals that differ in their developmental mode, but also in their geographic ancestry. So in fact, in that case, it's planktotropes from New Jersey and lesotropes from California, which is the population we use to do all of our QTL mapping. So that's just kind of a sample that was convenient. And so those two populations can distinguish based on spermatophores and choose mates assortatively, but we don't know if it's based on larval mode or some other thing that differs between California worms and New Jersey worms. So I think Christina's lab will solve that pretty soon. But it's clear that assortative mating should play a role here to prevent mismatch. And that leaves us with this kind of mystery of why the population genomic data look the way they do, where there's these really narrow FST peaks and most of the genome's totally indifferent to developmental mode. That implies lots of gene flow so that those peaks can become narrower and narrower over lots of recombination events over a long history. And so somehow there's lots of gene flow despite the fact that hybrids are maladapted, that we don't observe hybrids in nature, and that we see assortative mating. So yeah, there's lots of PopGen mysteries that remain. Thanks.
Thank you.
Do you think it makes a difference between the Newark mudflats where I grew up, where pollution is quite a problem, and California or even Rhode Island?
Yeah, I think that this is a fantastic model for ecotoxicology, and that probably a lot of geographic differentiation that we're going to find represents adaptation to local conditions. And so we saw that the lesotropes between Rhode Island and New Jersey are really genetically different from one another. And some of that could be local adaptation to dioxins in Newark Bay, for example. And so Connor is actually collecting a bunch more populations now where, in principle, we can kind of sort out some of those signals. But for sure, these animals are living in pristine and also incredibly polluted areas and probably adapting.
thinking about the paternal effects, do you think that could be related with the spermatophores containing nutrients? I know some spiders and other like spermatophore animals do that.
Yeah, I think that's absolutely a possibility. They structurally, they look like bundles of sperm. Like the sperm tails are actually exposed in those things, so it's not like a package that we sometimes associate with spermatophore. But I think it could nevertheless contain nutrition. The whole spermatophore is kind of the same size as a single oocyte in an elastotroph. So I don't think it can be like direct provisioning to oocytes, and that some kind of chemical signaling is more likely. But yeah, we need to find out what's in those things. Yeah.
Thank you for the great talk. So, seems there are some QTL. They have very different allele frequency in the cystotroph and planktotroph. And so I wonder whether they mate with each other very frequently, or they almost never mate with each other in nature.
Right. So in nature, we haven't observed intermediates, or by genotype we don't observe F1s or subsequent generations.
I see. So, but do they exist in the same location, like to?
Yeah, in a few locations they do. Yeah. So I'll say that the two locations where we've collected both of them in the same place, we found them both the first time we collected those places, and then we're like, this is fantastic. We're going to go back and collect 1,000 of them, and then after that, we only ever find the lestotrophs at those places. So it does seem like maybe it's a transient thing where migrants are coming in, and they're selected against, and then they go away again.
I see. So does that mean that they're under the way of speciation, like they're becoming two different species? One is having.
Yeah, I don't know. I mean, the genetic differentiation is really confined to specific bits, and most of the genome has gene flow. So I think it's a question for people who think about the semantics and the theory of speciation to answer your question. I don't know.
Okay, thank you.
Awesome. Let's thank Matt one more time, and then.